Harvard Medical School, and today I'm going to talk about a graph-based approach to diploid genome assembly, and this work was done as a PhD student in Max Planck Institute. So humans are diploids, and there exists two homologous copies of each chromosome encoded in these two colors. And sequencing technologies produce the genome sequences in pieces called as sequencing reads, and the reads contain errors. The goal here is to reconstruct the underlying genome sequences from the reads. And this process is called diploid genome assembly. It has several applications. It can be used in personalized medicine. It can be used to study human migration, evolutionary selection, and population structure. There are different approaches to genome assembly. The first one is reference-based haplotyping. So here you are given the reference genome and the reads from the target genome. And then you align these reads to the reference genome and partition the reads to two sets to get these two haplotypes. The main drawback of this approach is that since there is a reference genome involved, which leads to a reference bias, and it does not work for structure variants. There are tools for reference-based haplotyping such as WhatsApp, HapCut, and HapCall. So the next approach is de novo assembly, where we don't use the reference genome, instead construct the genome assembly directly from the reads. So the main idea here is you represent the set of reads as an assembly graph, and there are two types of assembly graphs. So given these reads, you break the reads into words of fixed length, for example 4 here, and each word is a node. And the connections are based on the overlaps between the node sequences, and this is a de Bruijn graph. The next one is the overlap graph, where you take the whole read as a node, and connections are based on the overlaps between the node sequences. So these are the two types of assembly graphs. And now if you, for the diploid genome, you see a special structures in these graphs. For example, bubbles here, which are defined as the set of disjoint paths that share the common start and the end node. And these bubbles represent the heterozygous sites over the diploid genomes. So now, given this graph, you can find our Eulerian or a Hamiltonian path to find a genome assembly. Now, the main drawback here is the underlying assumption is that the genome is a haploid genome, and it requires high coverage data set to generate a good quality assemblies. So there are tools for genome assembly such as Canoe, Spades, and so on. And Falcon Unzip is the only available diploid assembler. The basic idea of Falcon Unzip is it collapses the bubbles in the assembly graph and then align the PacBio reads to this collapsed consensus and find out the phase of each read. And the limitation here is that it cannot phase all type of SWIS and cannot work with different heterozygous rates. So here we propose our graph-based approach, where we work in the space of assembly graphs. So here is given the assembly graph and the bubbles that could represent SNV or SWIS and so on. And the linear ordering of these bubbles is called a bubble chain. And in each bubble, you see multiple allele paths here. And now we align the long reads to this assembly graph, and we want to find out the phase of each read, whether to identify the haplotype or of each read which is encoded in these two colors, and to get these two haplotypes. So The two main questions we ask here is, what is a cost-effective way of generating the good quality assemblies? Can we face even large structure variations over the genome? And here is our diploid assembly pipeline. We first construct an assembly graph from the Illumina data, so which is an accurate data and provides a good backbone for the subsequent steps. And now in the assembly graph, we remove the overlaps, and this process is called bluntification, which results in a sequence graph. The sequence graph, the nodes are the sequences, and the edges represents the adjacencies between the node sequences that they connect. And then we find out the bubble chains. Finding bubble chains is not a trivial problem because of the cycles and the branches in the graph. But the good news here is finding bubble chains is similar to solving the de novo assembly problem, and based on this notion, we can find out the bubble chains. And we align long read data sets such as PegBio to this assembly graph. Now, based on the PegBio alignments through these bubble chains, we want to find out the two haplotype paths in this graph such that we remain consistent with the reads. Now let's go more in detail how we can solve how we can phase the bubbles. So the main observation here is phasing bubble chains is similar to phasing multi-allelic variants. Therefore, we generalize the MEG formulation for reference-based haplotyping to multi-allelic variants. So let's consider this example where we are given the assembly graph and the reads aligned to it. And now, due to the sequencing errors present in the reads might align to the different alleles. Therefore, we want to flip the set of alleles with the minimum cost such that we can assign every read to one of the two haplotype paths. And for example, if we assume the flipping cost to other alleles is constant, which is 10, we can flip this allele to the other allele by paying a cost of 10, and then we can get the two haplotype paths encoded in these two colors. So now and this problem is called as GMAC problem. And now in order to solve GMAC, we use a WhatsApp style dynamic programming algorithm. So the main idea here is we find the optimal path pair of first k bubbles and then extend it to find for k plus 1th bubble with the resultant optimal path pair for first k plus 1 bubbles. And at every step, we remember the information only from the previous step. And we continue this process until the last bubble and backtrace to get the two haplotype paths. So now I'll illustrate this algorithm through an example. So first, let's consider a biallelic case where we have a bubble with only two alleles. And we want to find out the cost for the bipartition of the reads encoded in these two colors. So for this bipartition, we pay a cost of 10 if we flip this allele here, and for the blue partition, we don't pay any cost. So in this way, we can compute the cost for all possible bipartitions, and we store it in a DP column. Now for the multi-allelic case, we consider all possible path pairs out of the 4 alleles here and compute the bipartition score for every bipartition of the aligned reads. So in this, the possible path pairs will be 4 choose 2. Now in the recurrence step, for example, for the second bubble, we consider path pair out of 3 paths, and we compute a score for every bipartition of aligned reads, given the cost for all bipartition for the intersecting reads. So intersecting reads are those reads that cover the current column and the previous column. For example, here, R1 and R4 are the intersecting reads that cover the second bubble and the first bubble. And we compute the minimum score of all bipartitions for these intersecting reads over all possible path pairs and store it in a DP column. Now, for the second bubble and for the partition encoded in these colors, we first compute the initialization cost as we did before. And for the recurrence cost, we want that the bipartition for the intersecting reads remain consistent over the first and second bubble. For example, here R1 and R4 lie in a different partitions. Therefore, we consider the cost for this partition and add it here and store this cost in a DP table. And we compute this cost for all possible bipartitions and store it in a DP column for this bubble. And this way we can recurse until the last bubble and finally backtrace to get the haplotype paths. So WhatsApp algorithm is a fixed parameter tractable in maximum coverage and maximum number of alleles in a bubble. It remains polynomial in the number of bubbles and independent of the read length. Therefore, it's suitable for longer reads. Now, in order to evaluate the performance of our algorithm, we considered a pseudo-diploid genome by combining two yeast strains, that is SK1 and Y12. We considered Illumina and PegBio data, which is publicly available. As a ground truth, we considered assemblies that were generated using high coverage data set by considering each of these strains independently. Now, to answer our first question on what is the cost-effective way of generating the good quality assemblies, we downsampled the PegBio data to different coverages of 10x, 20x, and 30x. And we compare our approach to Falcon Unzip, which is the only available diploid assembler. And we see that as we increase the coverage, the average identity of the assemblies improves. And at 30x, we can deliver the assemblies with an average identity of around 99.8%. And whereas Falcon Unzip can deliver an average identity of around 99.4%. And Falcon Unzip is not designed for lower coverages, so there are no numbers here. And now if we compare these numbers here, we see that we can deliver assemblies with the average identity of 99.5% even at a lower coverage of 10x. So the main reason behind this, we use the Illumina data as a backbone, and also our model is good enough to correct the PegBio sequencing errors. Furthermore, the similar behavior is seen in the phasing error rate. We see that we can deliver the assemblies with the phasing error rate of around 0.4%. Now, to look into the completeness of the haplotypes, where the genome size is 24 megabase pairs, we see that at 30x coverage, we can generate near complete haplotypes, whereas Falcon Unzip can generate almost complete primary contig, but it cannot generate complete haplotics, therefore resulting in the assemblies of size 16.6 megabase pairs. So now to answer our second question on can we even phase structure variants, so we looked more closely into the haplotype sequences for every bubble, and we find out the difference between the allele size. And on the x-axis, you see the allele size difference, and on the y-axis, you see the number of bubbles. So in this histogram, you see that there are around 90 bubbles that contain SVs of longer than 20 base pairs. So this plot indicates that we can even phase structure variants. So to conclude, in this, we developed a novel haplotype-aware de novo assembly pipeline, And we show that through this integrative pipeline, we produce good quality haplotypes even at a low coverages of 10x. And we demonstrate that we can even face large structure variance. For graph alignment, see poster A367. Thanks.
We have time for a few questions.
Hi, great talk. I didn't quite catch it, but why even use the Illumina data at all? Why not just establish your backbone graph from the PacBio data as well?
So our interest was to generate the cost-effective assemblies. If we want, since as you know, the PacBio data is erroneous, and then we would require a very high coverage data set to reach to the level of good quality assemblies as we can do by using Illumina data.
Hi, I have two questions. The first question, do you consider any false positive of your assembly? The second question, have you tested on the genomes with more complexity compared to yeast?
Yeah, so for the first question, like the false positives of the structure variants, so we haven't yet looked deeply into the detailed analysis of our structure variants. And to answer your second question on, could you repeat this? We have been trying to run this pipeline on a more complex genomes, like a more heterozygous rate, but we are still in the process of doing it. Thanks.
Last question.
All right, so how are you handling variants such as repeat expansions where you're traversing the same part of the graph different numbers of times rather than having disjoint paths?
So right now, we don't face the repetitive regions. Based on the expected coverage, we first identify the repetitive regions and then just take them out, filtered.
Let's thank the speaker and thank you very much. Nice talk.